Welcome to the Chemistry, Biology, and Math Revision Hub. Today we are discussing the January 2021 Pearson Edexcel International A-Level Biology Unit 6. This paper is 50 marks. Let's go direct to question 1. Question 1 says, the photograph shows flowers of a water hyacinth plant. So here we see the plant they're looking at. And the question says these plants are an invasive species found in lakes and rivers throughout Asia. They grow rapidly, producing many flowers and seeds. A student observed that each flower has 6 anthers, and 1 stigma. Each anther was observed to release many pollen grains. When pollen grains from the water hyacinth are placed in a sucrose solution, they grow pollen tubes. You guys remember from AS Biology about the germination of the pollen tube. So the grain germinates and then the pollen tube is going to be formed. So this could be seen growing within 30 minutes. The student wanted to find out if an extract from the stigma stimulated the growth of the pollen tubes. So this is an investigation the student is trying to carry out. So they say, describe an experiment to investigate the effect of this extract on the rate of pollen tube growth. This extract is actually got from the stigma, and we're going to expose it to the pollen grains and see if germination will occur and the pollen tube will grow. So to perform experiments, of course, here we have to expose. I said perform experiments by exposing the pollen grains to the extract from the stigma, and then we are going to compare the pollen grains with pollen grains not exposed to the extract from the stigma. Basically, we have the value. The thing we have to measure here is the length of the pollen tube. So we need to use a microscope to be able to measure the length of the pollen tube grown every 30 seconds. Again, you remember 30 minutes. The student said 30 minutes for up to 12 hours. So minimum is going to be 30 minutes, but to see enough growth, we're going to do 12 hours. The control variables here, because again, pollen tube growth is going to be enzyme controlled. So we expect temperature and pH to be a requirement as conditions that have to be controlled. So we will control the temperature using a water bath, and we'll control pH using buffers. The age of the plant that we're getting the extract from, as well as the plant we're getting the anthers from, should be the same. Or the pollen grains, the part with the anthers that gives the pollen grain, should be from the same flower and therefore the same age, same species and everything. Then we can calculate the rate of pollen tube growth by dividing the change in the length of the pollen tube by the time it has taken for it to grow or to increase in size. We will repeat this experiment, treating with and without the extract from the stigma, and see the mean values that we can obtain. Those mean values will be the values we are looking for. So we would expect that if the extract increases the rate of growth, we would expect that those that were exposed to the extract will have longer height than those that were not exposed. So the next part says, the tip of the pollen tube is protected by an outer layer of pectin. Pectin is a polysaccharide. Starch grains, mitochondria, and vesicles are present in the tip of the pollen tube. Describe how these structures are used in the production of pectin at the tip of a pollen tube. Now, at the tip of the pollen tube, of course, there is going to be digestion. So starch is going to be digested or hydrolyzed to produce glucose. And as you know, glucose is used in respiration to produce ATP. So this glucose can be used to produce the polysaccharide by the condensation reaction of the glycosidic bonds. I don't know if you guys know, if we break down starch to produce glucose, some of the glucose could be used for ATP generation. However, some of the glucose could be polymerized to produce a polysaccharide, which is pectin. And of course, when we do produce a polysaccharide from glucose, there is going to be water loss. That is the condensation reaction due to loss of water as the polymers or as the glycosidic bonds are being formed. Then the produced pectin is packaged in vesicles. Remember, here we are looking for things like pectin, which is a polysaccharide, and then starch grains, basically mitochondria, as well as vesicles present at the tip. The starch grains are going to be used to break down, they're broken down to produce glucose, and then the glucose is going to be used to make the polysaccharide, which is going to be packaged into vesicles and the vesicles will fuse with the cell membrane, releasing the pectin by exocytosis, that is, okay, releasing it outside the cell. And then the mitochondria can use some of the glucose to provide energy. This energy is going to be ATP, because again, the process of making pectin is energy requiring. It means some of the glucose is going to be used to make pectin, whereas the other glucose is going to be used to produce ATP that is required to carry out the production of pectin, as well as the exocytosis and the energy required for enzyme activity in the process of making pectin. This brings us to the end of question 1. We will continue on to question 2. Question 2, the diagram shows some maggots in a respirometer. So here we see the colored solution. We would expect there is oxygen in here because we see soda lime here. Soda lime absorbs CO2. So it means all the CO2 produced by these maggots is going to be absorbed here. It will not penetrate to this side. So we will only have oxygen in this tube here, or in the capillary tube. And that can enable us to basically measure the volume, or be able to calculate the volume of oxygen that is present, or that is taken up as this. The organisms are respiring, the color solution is going to move this side because oxygen is being used up. So the distance is the liquid is going to move towards this side. So we can be able to use a specific formula and find the volume of oxygen used up. So they say this respirometer can be used to investigate the uptake of oxygen during the respiration of maggots. A student used this respirometer with soda lime to measure the movement of the colored liquid. The table shows the results. Here we have the different, the time in minutes as well as the distance movement of the colored solution or colored liquid. So the first question says calculate the mean rate of oxygen uptake. If we are looking for mean rate, it's going to be, of course, amount of volume of oxygen taken up within a specific period of time. So here we will get, they've given us the formula, which is pi r squared L. So they want us to give the answer into significant figures. To use this formula, we need to know radius, and radius is equal to diameter over 2, which is 0.2 divided by 2, giving us 0.1 millimeters. That is the radius, so you can see it there. After finding the radius, we also need to find the mean distance, or this mean distance per time. It means I'm going to get the total distance, that, adding up everything. And you know, the time in minutes was 5 minutes divided by time in order for me to give me 44 millimeters per minute. So basically 44 millimeters per minute is the mean distance per time that I have. So I can be able to find the volume per time as V is pi r squared L, which is the r here squared, and then the L, which is the length, the mean length moved within per minute basically. And when I multiply, I get 0.138. However, we need to present our answer into significant figures, so round this off to that, we get 0.14 millimeters cubed per minute. The next part says, the mean rate of carbon dioxide released by the maggots is 0.11 millimeters cubed per minute. Calculate the respiratory quotient, RQ value for this investigation. You need to remember that RQ is equal to volume of CO2 produced divided by the volume of oxygen used. So we know from here that the CO2 produced is 0.11 because it's given. And from our previous question, we have calculated that the volume of oxygen used up is 0.14. So when we divide the two, we get that as our RQ. Next part says, the soda lime was removed and replaced with glass beads in order to calculate the RQ. Suggest why this allows the student to calculate RQ. Again, you remember we said if you're carrying out an experiment like about respiration in a respirometer, the volume of oxygen is going to be the only gas within the capillary tube. So, however, all the CO2 produced is going to be absorbed by the soda lime. So it means we can get the soda lime and use it to determine the volume of carbon dioxide that has been released. So this soda lime absorbs CO2. It is therefore removed in order to predict the volume of CO2 absorbed. And since we find the volume of CO2, we can be able to calculate the RQ for the experiment. The next part says, set two are biotic factors or biotic variables. Here, the biotic means living or biological, basically living. State two biotic variables that could affect this investigation. When they ask you to state biotic variables, you're looking about maybe the mass, to do with the organisms that you're working with, the age, the mass. So here I say the first variable should be mass of the maggot. You could talk about species of the maggot, the age of the maggot, the sex of the maggot. All those are going to be biotic factors or biotic variables. So they say choose one of the variables you have identified in B1, and then we describe how this variable could be controlled and the effect it could have on the results if it is not controlled. The species of the maggot should be controlled. If it's not controlled, in this case we can see it can be controlled by ensuring that they are all coming from the same species or even from the same parent. There we can guarantee that they are the same species. And then if this is not controlled, then the results are not going to be valid because the different maggots are going to have different rates of respiration, and therefore they will use up oxygen or produce carbon dioxide at different rates in their individual different experiments, leading to invalid results, and therefore the oxygen consumption or carbon dioxide release will not be the same. Our results will not be consistent. So this brings us to the end of question 2. Let's go to question 3. Question 3. People living in some villages in Ecuador, a South American country, keep chickens as a source of food. These people have been encouraged to add ampicillin, an antibiotic, to the chicken food. Feeding ampicillin to chickens may cause the bacteria E. coli to be resistant to ampicillin. Village A was the first in the area to add ampicillin to the chicken food, and Village B has only recently added ampicillin to the chicken food. Samples of E. coli were taken from the guts of chickens from each village. Each sample was cultured on a nutrient agar. You guys know agar plates. A disc containing ampicillin was placed on each culture. After incubation, the diameter of the clear zone of inhibition was measured in millimeters. The photograph shows the zone of inhibition on one nutrient agar plate. So basically, this is where this area, the zone of inhibition, it means there will be no growth of the specific microorganism. Let's go to the next part. So here we have the results. I marked the results with the greatest as well as the smallest per village. This is village A, and that is the data for village B. So they say state a suitable null hypothesis for this investigation. This null hypothesis, because it's comparing the results of two different areas, and the results are basically the diameters of the area or of the zone of inhibition, I say there is no significant difference between the mean diameter of the zone of inhibition between the samples from village A and village B. There should be no significant difference, and then we can see whether that null hypothesis holds or not. Part B says, draw a suitable table to display the results and calculate means for village A and village B. So here I presented the results in their raw manner. So you can see the diameter of the zone of inhibition in millimeters. We see for village A and we see for village B. All of them were placed, and then I added up everything and divided by the number of samples. Again, here to count: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So you add up everything and divide by 15. You will get this as the mean for village A and that as the mean for village B. So as you already know, the next part is going to be about you drawing the graph and representing the data. So here we see, I'm going to make this small so that you can see my answer properly. So they say plot a graph to show the mean diameter of the zone of inhibition for village A and for village B, and they say include an indication of variability. It means they want you to include the error bars, variability of the results. So as you can see here, I put the data x-axis, the village. So we have the data for village A and the data for village B. Here I put the diameter of the zone of inhibition in millimeters, and then the mean value is here for village A, and for village B you can see 17.3 is there. Then the higher bar shows us the highest value for village B, and the lower 14.8 is the lowest value for village B. 14.4 is the lowest value or lowest diameter for village A, and 18.2 is the highest diameter for village B. And again, to show you, here the error bars do overlap. I think you can see that, you can note it. They do overlap. So maybe later when we're trying to make a conclusion, you can keep that in mind. A student analyzed the data with a t-test using the formula. So we have the formula given here. This is the t-test. So they say where x bar is the mean value for each village and n is the number of samples for each village. They've given us the ss squared and sb squared. They want us to substitute. Since this is given, that is given, this we already know, and that we already know because we know the numbers of the samples, we can be able to find all the other values basically, which is easy. We also know that and that. So here I came and say XA, which of course is 17.3 from the previous data, and XB was 16.2, divide by the root of 0.99 given to us, and that over 15. Remember, each village had 15 samples, so we can be able to find the value as 2.76. This 2.76 is the calculated value. It's the calculated value of t. So let us go to the next part. It says the table shows the critical values of t for different degrees of freedom. Now again, remember 15 degrees of freedom. In this case, degrees of freedom, if this is 15 minus 1 plus 15 minus 1, it means the total degree of freedom is 28. So 28 degree of freedom, or the numbers of degree of freedom, 28. So you will come to your table of critical results and look for where 28 is, and then you determine the value or the critical value at p is equal to 0.05, and this critical value is going to be 2.05. So that is the critical value we have. So the next part here, I'm going to let you to those of you who want to read this part, just to show you, but I'm not going to talk about it now because it's just simple notes I put there. So they're going to say, a scientist suggested that there was evidence for the development of resistance to ampicillin in E. coli in village B, criticized this suggestion, and used the information given and analysis of the results to support your answer. So you can see that the critical value was 2.05, the other calculated value was 2.75. So based on these results, this value here, which is the calculated value, is greater than the critical value, so the null hypothesis should be rejected. The null hypothesis, remember, said that there is no significant difference between the diameter of the zone of inhibition between the two villages, so it should be rejected. Meaning there is significant difference. So bacteria from village A had more resistance to antibiotics since their mean diameter of the zone of inhibition was smaller than those in village B. And again, village B has just recently been added because antibiotics have just been recently added to village B. So their chickens, and the diameter of the zone of inhibition was big. That could be made, we could conclude that there was lack of a fair data or fair comparison, and therefore this data may not be that reliable due to the differences. The other chickens in village A have been taking antibiotics for a longer period of time, so there is a higher chance that there is more resistance than in the newer village in village B who have just introduced the antibiotics. So the next part says suggests 2 improvements that could be made to this investigation. We need to control all the abiotic factors, they have to be the same types of chicken, they have to be the same sex, they have to be the same age, and then we have to sample more villages, or to make it fair, we have to use more samples, and then the results have to be compared with other villages that are using maybe different types of antibiotics, so that we can conclude that actually there is antibiotic resistance that is going on. This brings us to the end of question 3. Let's move on to question 4. Question 4, the photograph shows a section through a wheat grain. So in the wheat grain, we see the embryo and we see the endosperm. So the first part says, wheat grain store starch in the endosperm. During germination, the embryo releases amylase, this is an enzyme, from the cells surrounding the endosperm. The starch is digested by the amylase. This allows the embryo to grow. Of course, starch is going to be converted into glucose, and the glucose is going to be used as a source of ATP, which provides energy for the endosperm for the embryo to germinate. So abscisic acid inhibits germination of wheat grains. A student formed the following hypothesis. The greater the concentration of abscisic acid, the smaller the production of amylase by the wheat grain. Plan an investigation to find evidence to support or reject the null hypothesis. So here we're going to vary the concentration of abscisic acid. As you can see, it's going to be the independent variable. So let's see, we need to do some preliminary experiment. Describe preliminary practical work that you must undertake to ensure your proposed method would provide quantitative results. Here we have to find suitable concentrations of ABA we're going to use. Remember, I've told you the independent variable is the concentration of ABA. So we need to find which suitable concentrations are we going to use. Are you going to use 1.0, 2.0, 3.0, and so on until 5.0, or you're going to use 0.5, 1.0, 1.5, and so on. You have to determine which ranges of concentration you're going to use. And then we have to find a suitable method to measure starch digestion. Starch is going to be converted into glucose. How can we know the amount of starch that is broken down or the amount of glucose that has been digested, that has been produced by the amylase in the digestion of starch? So we also have to find a suitable time scale for starch digestion to begin. And then lastly, we have to find the optimum temperature, optimum pH, and for the reaction to occur or for the digestion to occur. So next part says, devise a detailed method including how you would control and monitor the important variables. Here, the dependent variable is the size of the clear area, meaning the area that has been digested, area that amylase has been broken down or that has been digested. And then here what we have to do, we have to wash the endosperm or rinse it in a sterile liquid like sodium hypochlorite. Here we will ensure that any germination or any glucose that is being produced is actually not used up by other microorganisms, leaving us with a less concentration of glucose than we actually should have. So conditions have to be sterile. So this removes any microorganisms present, and then we will soak the endosperm in concentrated ABA. Remember, ABA, they say ABA inhibits, so soak it into different concentrations of ABA. This is the independent variable, the variation in the concentrations of ABA. So ABA is abscisic acid. Then we'll place the endosperms onto our starch agar. Remember, endosperms that are not inhibited are going to release amylase, and the amylase release is going to digest the starch onto the present in the agar. So if ABA is a good inhibitor, it is going to prevent the endosperm from releasing the amylase, and that means there is going to be less or no amylase being released onto the starch agar, and therefore there will be less area that is digested, and therefore the size of the clear area will be smaller. So here we say the 5 different concentrations of ABA can be like 0.5, 1.0, 1.5, 2.0, and 2.5 can be used, or you could say 0.1 and so on, can be used to soak the endosperm. And then we will incubate the agar at room temperature for about 24 to 72 hours. You want to give them enough time so that it's easy, it's clear, you can clearly see the clear zone. Then we will measure the diameter of the clear zone to determine the digested area of the starch agar. And then we will use endosperms from the same species to make sure the biotic factors are controlled. Same species, same age, and everything to be the same. And then we'll control the temperature using a water bath and control pH using a buffer. And we have to control maybe starch present within the agar so that we do not have so many variations. Lastly, we have to repeat the experiment with at each concentration of ABA. Remember, the concentration you have chosen is from 0.1 to 2.0. It means at each concentration we gotta carry out repeats in order to calculate the mean, and then we will use that mean value as a representative value for that specific concentration. Describe how your results would be recorded, presented, and analyzed in order to draw conclusions from your investigation. So here, after we obtain our results, they are going to be mean values because we are going to do repeats at each concentration of ABA and find the mean value. So we'll get those mean values and tabulate them, put them in a table, and then the table should have headings and units. Then, referring to the table, we will draw a graph. It could be a scatter diagram, or we could draw a line of best fit, or it could be a line diagram. In this case, after we can use the data, this graph is going to have, of course, labels as well as units, and we can use appropriate statistical tests like Spearman's rank correlation or Pearson's, and then we can be able to make a conclusion about the obtained results to see if there is significance or not, or if the student's idea or theory was right or not. Lastly, suggest two limitations of your proposed method. The proposed method is going to be kind of tricky because it's going to be difficult to measure the areas that are too small. I know some ABA is going to have some tiny inhibition, maybe in smaller concentrations, and we will expect the diameter of the zone of inhibition is going to be really, really small to be measured, so that is going to be a difficult thing to do. And then there is going to be some difficulty in maintaining aseptic conditions because, of course, you have to wash the seeds. What if there are microorganisms that are going to remain behind? That is going to be tricky. And then some growth regulators could be present. Now, we know release of amylase could not be only controlled by one thing, or even the amount of amylase present could be different. So it means some seed could release more and another could release less. So those genetic or related things or the differences in genes could lead to different seeds having different productions of amylase, and that can lead to different amounts of digestion of the starch agar, meaning our results are going to not be valid or not be representative enough due to the differences of the differences within the seeds that we have used, basically. This brings us to the end of question 4 as well as the end of this paper. Thank you for being with us. I will see you in our next paper. Goodbye. Don't forget to subscribe to our chemistry, biology, and math revision channel. I will see you in the next video.